I've been very interested really in working in Parkinson's disease all of my career. And what most of us are working on are the movement component of the disease. We now know it's much more multifactorial, but actually it's the movement that is one of the major problems. And the movement's caused by the loss of the nerve cells sitting deep down here in the midbrain in a region called the substantia nigra. And they send their processes up to these other regions and release their dopamine, and that's what causes movement. And it's when those cells are lost that all the movement problems come in Parkinson's patients. What we do know is if you put a transplant back into this area here and it releases some chemical dopamine, then actually the patients show much more improvement. And there have been about 300 or 400 clinical transplants that have been carried out in the world up until now that have shown proofs of concept that if you put cells back in here, they can give improvement to some Parkinson's patients. And actually there's some clinical trials going on now. I think the first patients are being transplanted in Cambridge this year in a new cross-European consortium. So there is proof of principle that if you can put some cells in here, they can release the chemical dopamine. If they're nerve cells, they connect up a bit with the host brain. Actually, you can have a lot of improvement. But we can't give this out as a therapy for all patients with Parkinson's. One of the biggest problems is that to do a transplant successfully, you might need 6 or 8 embryos to take out the tissue to transplant into those patients. And you can see that's huge moral and ethical issues involved with collecting that much tissue. So what the world of research has gone into looking at Parkinson's is, can we use stem cells instead for our therapies? Now, I think probably a lot of you read about stem cells, and a lot of the literature out there, a lot of the media is quite confusing. There's not one type of stem cell, there are many types. And this is just to show you, probably not an inclusive range, but different types of stem cells that we are trying for research. So you can get stem cells out of the very early embryo. This is an embryo that's only 5 or 6 days old. And these blue cells sitting here in the middle, they can form embryonic stem cells. So that's one cell type that we use in our lab, and you'll see some of that in my experiments. Then a bit later, as the brain's developing, this is now an embryo brain, you can take out neural or brain stem cells. Us as adults, we have plenty of stem cells still in us, thankfully. It doesn't matter how old we get. And you can find them in the blood, the bone marrow, the skin, and importantly, in a couple of places in the brain as well. So we could use those as a source. And then the other stem cells that have been really popular in really the last 10 years are cells that are called induced pluripotent stem cells, or IPS cells. And these basically are made by taking a piece of tissue, you can take this from an adult, and actually giving the tissue some factor and making those stems change back into stem cells. So there are all kinds of stem cells out there, and one of the big questions is really what to do with them. They're quite easy to take out. The big thing about stem cells is they like to divide. So you can take them out. These are the embryonic stem cells again. You can take out these blue cells, put them in a dish, and they make these little colonies. Here's one growing here. This is one we've grown in the lab. And they do this really easily. So we can make lots of them. So we could treat lots and lots of people with these cells. We give them lots of food and nutrients. We feed them. They have to be covered in liquid, and this liquid contains sugars and salts. It's just about like we have to have 3 meals a day. Our stem cells have to have lots of food as well. And we also give them some extra goodies in here to help them to keep dividing and to keep them happy and healthy. But the biggest challenge is, once you've got your stem cells, how do you get them to be a therapy for Parkinson's? And I see the challenge as 3 main things. The first thing is you have to convert your stem cell into a nerve cell. Our best transplants will work if we have nerve cells in our transplants, because they will connect with the host brain nerve cells, and actually having that connection means you get controlled release of dopamine, of the chemical. So that's the second thing we want. We don't want just on nerve cells. There's lots of different types of nerve cells in our brains. They make all different kinds of chemicals. They do all different kinds of jobs. Actually, what we want is just nerve cells that make dopamine. And the last thing, and also this is a very important thing, and I think this is why stem cells really haven't got into clinical trials yet for many diseases, is we have to control those cells if we put them into somebody's brain. So we don't particularly want any dividing cells in there. And stem cells, their job is to divide. So that really means we need to convert all of our stem cells into a cell that won't divide. And that's quite a huge safety issue. So that's really, these are really the things that are holding the field back at the moment. So my lab's been interested in tackling some of these challenges, and particularly the first one, converting stem cells into dopamine nerve cells, or the first 2, if you like, of those challenges. So the problem is it's not very efficient. You've got all those lovely cells in your dish, and actually you try and convert them, and you maybe get 1 or 2%, 5 or 10% if you're lucky converting. The rest don't do it. And the way we think cells do convert themselves naturally as the brain develops is either they have genes inside the cells that are like a pre-program, if you like. They're on a pathway, and they know what they're going to do. They're going to switch on these genes to make nerve cells. Or actually, there might be something out there in the environment that makes this stem cell here become a very complicated nerve cell. And many groups are working on these here. My group, and I decided I wanted to look at this. What happens in our developing brain that might give us some ideas of how to put our stem cells in a culture dish and make them behave like the developing brain, if you like. So our solution was actually to go and look for some signals. Are there some signals out here that we can find, new ones that people didn't know about? And actually, would that help us when we're using our stem cells in the laboratory? So I had a very talented postdoc, Rowan, who started this project. And he said, okay, well, let's just work out where those signals might come from. So he was working with rats, and he took tissue. And basically, pregnancy in a rat's quite quick, a bit quicker than us, 9 months. Pregnancy in a rat only takes 21 days. And actually, the nerve cells that we're interested in are born around here, between days 12 and day 14, in that area there. So we know when those dopamine nerve cells appear. That's great. So what Rowan did is he took developing brain, he cut out the piece of tissue. Where this red dot is where those dopamine nerve cells are going to be born. So he cut out those pieces of tissue from different ages. Here's a 10-day embryo, here's an 11-day embryo, and here's a 12-day embryo. And that's just a piece of tissue he's grown in a culture dish. And what we've done is we've stained them with a brown marker that stains all the nerve cells that make dopamine. So hopefully you can see this one's white. Hopefully that's not brown, okay? There isn't any brown staining there, which means there's no dopamine nerve cells there at day 10. Here you can see some brown, hopefully, and there's a couple of bits here where the cell bodies are, and these are all the processes they're making. And at this one, at day 12, there's lots and lots of cells. So we know now the piece of tissue we need, and we know at day 12 is probably the best day for it. But what Rowan then did was a bit more smart. He said, okay, there must be signals in this piece of tissue that tells those cells to become nerve cells and dopamine nerve cells. So he dissected out pieces of tissue from this region, from different age embryos, and he went inside the cells, and he found all the molecules, all the proteins inside the cells that were in those pieces of tissue. We have some very lovely fancy equipment at Keele. This is a mass spectrometer. It's a Maldi Toff for those who know about these kind of things. And it basically, to explain to you how it works, is if you feed in a solution of proteins into the system, you can find out specific amounts of your proteins you're interested in. It'll identify every one. So if you were to take the equivalent of 120 kilograms of sugar and pour it into a big pile in the middle of the room, you could find 1 grain of salt in there if you threw it in. That's how sensitive these machines are here. So we could look for very tiny amounts of things that we thought might be important. So we did this. We chopped up tissue. We found all the proteins. And we found about 3,000. So we had quite a lot. So suddenly we didn't quite know where to start. So we thought, okay, well, we'll be a bit clever. We'll have a look and see what things we think might be important. And a lot of the signals we found were related to vitamins. So we thought, oh, this is interesting. A lot of the proteins we found were actually proteins that sit on the outside of a cell. Here's a cell here. And they're a bit like satellite receiver dishes, if you like. We call them receptors. And they respond to vitamins. So if there's some vitamin lurking around nearby, those vitamins can actually connect with these receptors. And that will trigger an event in the cell and will change what the cell does. And because we found a lot of these receptors, we thought, okay, well, the vitamins must be interesting. And so this is the line we've kind of pursued since then. So the first one we found, and I'll talk to you about 2 vitamins today. First one we found was the receptor for vitamin D. So we're very interested in vitamin D. It wasn't really known to be linked with Parkinson's that much at the time, but it's known to be very important in development, very good for bone health. So vitamin D is associated with bone. So if you get conditions like rickets when you don't have enough vitamin D. Great for an immune system, And there's been a possible link with multiple sclerosis as well. So it looked interesting. So what Rowan did was to say, okay, where is this vitamin D receptor, first of all? Let's just check it's in the right place. And a lot of things we'll do, and I'll hopefully show you some nice coloured pictures. We do a lot of staining of pieces of tissue or staining of our cells that we've grown. And we stain them with fluorescent coloured markers so that we can see them. And those markers show us what kind of cells they are. And then we've got lovely fancy microscopes. And these have been bought with funds in the previous years from Parkinson's UK, so thank you very much. And that allows us to see exactly what our cells are doing. This is an image that's created from this camera here. So first of all, Rowan looked to see where the vitamin D receptor was made. And this is a piece of developing brain, and you can see here there's blue here, which is all the cells. There's some green here, which is the vitamin D receptor. And there's red, which shows you specifically just the dopamine nerve cells. And you can see those are overlapping. So we knew the receptor was right in the right place. It's on the cells that are becoming dopamine nerve cells. So we got really excited about this. We thought, okay, we've got something here. So what Rowan did then is he took some cells and had a look to see what vitamin D does. You'll see this picture a few times. So it basically explains to you how you get from being a stem cell to being a nerve cell right over here, a complicated one with lots of processes. So here's a typical mature nerve cell, and here are the stem cells. You can see they don't go straight there. There's a lot of steps on the way. And I've drawn the two major ones. They go through a phase where they become specialized and become brain cells. So they can still divide, but they're brain cells now. And then they come to a stage where they stop dividing and become young nerve cells. And then they have to mature. We thought the vitamin D receptor was expressed right near the end here. So we thought vitamin D might be important up in this end of the process. So Rowan actually grew cells down in this end of the process. He grew some of these young nerve cells. And the first thing he did, he just added vitamin D to his cultures. So he took out these young nerve cells, he grew them, and he added some vitamin D. And you can see on the left panels here, this is a normal culture. It's got 4 colours again. You've got the 3 different colours and an overlay. And the interesting one here is the red. That's the dopamine nerve cells. So that's our controls with no vitamin D. And here, when you add some vitamin D, you can hopefully see there's a lot more cells. So that was really exciting. And he was very good. He looked at different doses. This is increasing doses of vitamin D up here. And he counted his total number of cells. And you can see from the controls, which are about 6% here, he's up to at least double, about 12%. So this was the best concentration he found, so he worked with that afterwards. But definitely adding vitamin D into the cultures changed everything. It made more dopamine nerve cells. Okay, so then we knew that there were more cells on our dish. What we didn't know is that because we're making new nerve cells, or is that actually because we're helping the cells that are there survive better? So he did an experiment where he looked at the dead cells in the cultures. So he added vitamin D to his cultures, and here's the nerve cells, and then he marked dead cells. You can see the red marker. There's a few little dots here, but not very many. Let me show you the compare picture. And you can see here, for the same number of nerve cells, there's a lot more dead cells in here. And what he'd done in these cultures, he'd added some vitamin D, but then he'd blocked it working. He'd stopped it working. He put a block in there. And so we now thought vitamin D now really stops cells from dying. So it's a survival factor, we think. And then we went a bit further and said, okay, how might it be working? How can vitamin D help nerve cells survive, and how might that be useful to us? What he did is he had a look through the literature, and he found a paper that said that vitamin D might help to make this special factor, glial-derived neurotrophic factor, GDNF. And so he said, okay, well, if I put vitamin D in my cell cultures, do I get more GDNF? And that's basically what he did. He took the liquid on the top of his cells in culture, and he looked for how much GDNF might be in there. And in the cultures where he had his vitamin D, he got a large amount. In the cultures without vitamin D, he got a lot less. And that's just showing you how it's quantified there. So it looks like by adding vitamin D on top of these cells, they suddenly start making GDNF. And that's really exciting to us. Here's the highly technical bit. This is to show you that this is a control culture, and you add the vitamin D and you get more neurons. You've seen that data before. If you block GDNF, but you've got vitamin D in there, that's this one here, you get a lot less neurons. So basically, we know that the vitamin D is making GDNF, and if we block the GDNF, we get less surviving neurons. So definitely our vitamin D is working by making more GDNF, and that's helping our neurons survive. What I haven't got here yet is the next step. We've started the next step of these studies. This is all done in cells in culture. We've now looked at whether vitamin D can help dopamine nerve cells in a rat model of Parkinson's disease. And that's work that we've been doing in collaboration with a group at Cardiff with Professor Steve Dunnett. And actually we've got some interesting data that suggests that if you give animals vitamin D in the drinking water, actually it can protect their nerve cells from an insult that we give them, a toxic insult or a toxin we inject. It stops as many of their nerve cells dying. So we've got some evidence that it might work in the animal models as well. So our conclusions are that we have the receptor in the right place, the vitamin D receptor, and it increases the number of nerve cells in our cultures. We know it upregulates this fantastic molecule GDNF, which we know is great for Parkinson's. And we know that's how it protects the dopamine nerve cells through this GDNF. So hopefully this is something very exciting. GDNF at the moment has to be delivered into the person's brain directly. So it has to be put in with a metal cannula. If we found something like a vitamin or a drug that can stimulate that happening normally, that would be fantastic. We don't know whether that happens yet, but we're really hopeful. We need to go and find out whether that does. That could make all the difference. So our vitamin D, we think, is acting at the end here, acting on our young or even mature nerve cells and making them much better. So it might be that actually we can use vitamin D on its own. We don't need to use it with stem cells. So while we were doing all of this, a colleague, who's now become a colleague, Professor Adrian Williams, he came up to us and he said, "Have you thought about vitamin B?" And we said, "No." And he said, "Well, I think it might be quite interesting. I think it might be involved down in these early steps here." So we said, "Okay." And after a few years of trying to find funding, we funded a lovely PhD student to do a project looking specifically at vitamin B. We thought we're going to have a look and see what that does. So we used this model, and we can grow all these cell types in our lab, but Sheila decided she'd start with these cells. So she was growing these stem cells in a culture dish, and she was going to look at what happened when she added vitamin D to these cultures. And she had a 14-day method to get from big stem cells to get to being mature nerve cells. And so she added vitamin B early on, either up to day 4 or up to day 7, and then had a look at what the cells became at the end. So here's the first results that she got. If she added her vitamin B to her cultures in the early days, day 0 to day 7, she saw that many more of her mouse cells became nerve cells. So here you've got all the cells again labeled in blue, and the green shows you the nerve cells. So this is a normal culture, just an average culture. We've got a few green cells in there. And here you can see at her top dose, which is up here, she got a lot more nerve cells. She also did a dose response, if you like, so she looked at increasing doses, and importantly, she saw that too much vitamin B is also bad. So there were no surviving cells in those cultures. It actually killed all the cells. So there is this thing, and it's likely to happen with all the vitamins, that there is a good effect at some doses, but you can have too much of a good thing as well. So what she's found out, and I've got a few slides just to show you this again, is actually what it does, vitamin B, it seems to accelerate everything. It's like pushing your foot on the accelerator. It's pushing those stem cells much faster to get towards nerve cells. And the way we found this out is we used a cell line, which we got from a collaborator in Cardiff, Professor Meng Li, and she's got cells that glow green. And they glow green at a certain stage. They glow green when they become brain cells, the dividing brain cells. So that's because they've got this SOX1 GFP marker. So here's cells that, this is taken now at day 4. So she added her vitamin B to day 4 and took the cells. There aren't many green ones there in that control culture. When she added vitamin B, she got a lot more green cells. So she can see by day 4, there's a lot more cells going a lot faster. They've got to become brain cells already by day 4, much faster with her vitamin B. And this is just 3 ways of showing the same thing. So here, she looked at a marker for stem cells and saw that the bottom panel here has got vitamin B. This is the controls. There's less of these red ones. There's less stem cells. and she saw that she had more brain cells. And here, and what she did then, so this panel shows actually her cultures she'd taken to 14 days. So she'd given them vitamin B for 7 days and let them go on for another 7 days, so they're at 14 days. And actually at 14 days, you get a lot less green ones because they've gone through this phase and on out to the other side. And in fact, that's shown here. So here she's got lots more nerve cells. So at 14 days in her cultures, she's got less stem cells, less dividing brain cells, and more nerve cells. So hopefully that's addressed the three things that we're looking for. We're looking for more nerve cells, but we're also looking for safer stem cells. Okay, the million-dollar question is whether we've got dopamine nerve cells. So just one more bit of data before that. This is to see that the stem cells are stopping dividing. She wanted to prove what's happening in her cultures. So she looked for how many of her cells are dividing, and because what she noticed was when she added vitamin B, she got many less cells. And actually she found it wasn't because the cells were dying. Vitamin B is not killing cells at all. It's actually that she's getting more cells because the cells are dividing. So the last thing she has done so far, before her PhD is ended, is to say, okay, these nerve cells, it's okay making lots of nerve cells, but actually we want dopamine nerve cells. And so she looked at her cultures to see how many dopamine nerve cells she got in there, and they're the red ones. She looked into 2 different conditions. In one condition, she got 5 times as many dopamine nerve cells. In another condition, she got twice as many. And this protocol she used here, this method, is one of the best methods out there for making dopamine nerve cells. So she's actually increased that by adding vitamin B to that method. So that's really exciting to us. One simple molecule has really changed our cultures. It's made us make a lot more dopamine nerve cells. So we're very, very excited about this. The picture down the bottom here just shows that those dopamine nerve cells actually have longer processes. So those black bars measure the average length of the processes coming out the cells. So we're making more mature nerve cells with our vitamin B. So where's that vitamin B working? We think it's working down here. It's working at this early stages here to move the stem cells much faster through this pathway to get them out this end. So that's very exciting data. And we haven't got that far with this yet. This is a new project, and then we need to take that and look at that further to see if that happens in the developing brain as opposed to just stem cells in our dish. But it might be a good way of converting our stem cells to much more safe nerve cells. So that's really all I want to say about the vitamins. We've looked at 2, and they look to be important at 2 ends of the spectrum. They both might be important to help stem cell therapies, but vitamin D might be important in its own right. And one bigger question, which we haven't yet addressed, is whether if we took vitamin B and looked at how the normal brain develops, maybe we should be thinking about vitamin B in pregnancy in the same way as we think about folic acid, giving that to pregnant females, pregnant ladies. Does vitamin B have a similar effect? Does it help the brain develop much more normally? Does a lack of vitamin B in pregnancy affect what might happen a lot later on? Can we prevent Parkinson's in any way? We have no idea whether they're connected yet, but that's an interesting line to follow, I think. So hopefully I've given you a bit of evidence that we can use vitamins to develop stem cell therapies.
Stem cells are really exciting cells for us because they are the basic cells of the body. We're really interested in using these cells because we can make them into the dopamine cells that are lost in Parkinson's disease.
So we're not really trying to rebuild the brain in a dish, but we are trying to rebuild the circuit that's involved in Parkinson's disease. Can we use this model to understand how we could rebuild the circuits?